In this video, we will talk about data visualization in R. You will learn about the ggplot2 package, which is a really powerful package that gives you the opportunity to design the most elegant and most versatile plots in R. So with that, here are the learning goals. So we'll delve more into ggplot2, but we'll start with producing a simple scatter plot with a geometry which is called geom_point, which produces scatter plots. And then we'll get into different geometries where we produce different types of charts based on the different types of data sets that we have, or the different types of variables we might have. We will also try to understand how do we visualize the distributions of a single variable, and how do we make sense of the relationships between two variables based on the type of those variables. And finally, we'll close by understanding how to save a plot and give you some troubleshooting tips and even some resources. So with that, let's just get into our prerequisites. So to follow along with this lab, it's essential that you install the ggplot2 package, the Palmer Penguins package, and the ggthemes package. And it's also essential that you load all of these. So what does that mean? Let's copy this here and go back to R. So here you can start a script if you're practicing in R, and you can paste this code. Now, if you already have installed these packages, you don't need to install them again. But let's say you hadn't installed these packages, you're going to run this one time, and you're going to see that this is executing. And now it's executed because in the console, we see this blue prompt again. And we can go back to loading our packages. So we can go back and load our packages here, and we're going to run these lines of code here. So here we're going to see some conflicts, but don't worry about that just yet. So basically, if you've done all this, you can then comment out line 3 of the code, so that every time you run this lab or anything that's going on in this lab, you can just forget everything else here, and this can remain as a comment because you've installed those packages. But remember that every time you're starting a session or you're opening this file, you would have to load these libraries because it does tell R these are the functions you would like to use. Okay, so tidyverse is something that we will use pretty frequently throughout the course, but today we're going to be talking about Palmer Penguins. This is a data set. It's a fun data set. We'll see what it contains, and we will also use ggthemes that allows us to do some site kind of styling that is that makes our charts better. Okay, so with that, let's go back. Now, you can take a look at the data set that we have from Palmer Penguins. The data frame itself is called Penguins, and that was collected by Dr. Kristen Gorman, as well as the Palmer Station Long-Term Ecological Research Program. And this data set is interesting. It has data on penguins in Antarctica. And there is information on the 3 species of penguins. There is the Adelie penguin, the chinstrap, and the gentoo penguins. And these are penguins that were observed on the islands near the Palmer Station in the Palmer Archipelago. And so if you want to learn more about it, you can always look at the help menu here and learn about the Palmer penguins. I'll just type this in your console and you can learn more about it. But I like the pictures. So here is the Adelie penguin on the far left, as you can see. This looks like this type of penguin this species has like a tuxedo on, a black tuxedo on. Maybe it's ready to go party. And this one in the middle called the gentoo penguin has like an orange or a red bill. And there is the chin strap penguin, as you can see here, the name says it all, right? So we want to explore this data set. So what we can do is take a look at just the first 6 rows of the data, and we can see here that there are 8 columns, right? So heading the data by default gives you just the first 6 rows of the data, and here we can see that there's information on the species, right? The 3 species that we have, the island to which the penguins belong, the length of the bill, and also the flipper length, as well as the body mass of these penguins. Okay, so that's the first thing you can do, is take a look at that. And then we can also see what are the dimensions of these of our data set called penguins. This is this has 344 rows and 8 columns. Okay, now you can also achieve that by by doing glimpse on penguins, right? This is pretty interesting. So let's just go back for a second to R, and we can type in glimpse penguins. And if we run this, you will notice this is very similar to what we were doing with structure, right? It's sort of giving you this in a different format. Here it is actually specifying the number of rows and the number of columns, and it is telling you all of the variables that you have in columns, right? So if you have this, we can load this data as well if you like if you just write data penguins, this is going to load the data set on the right hand side. So let's go ahead and run this, and here it's saying that oops, there are 2 penguins data sets that got um, loaded here, but the one we want is this first one here, which is just called penguins, not penguins underscore something, but just penguins. But this is this one is, I believe, more detailed, so we don't need that one, but this is the one we want. Okay, so we don't want the raw file, we just want the penguin file. Okay, so this is how you can work with it in R. So let's go back here. Now you know how to glimpse the data. This is a function that is from the dplyr package in the tidyverse, which is in the tidyverse packages. But it's very similar to the structure command in the base R, that you know about. Okay, so let's move forward and try to see if we know that these are the different types of variables available to us, what are the types of questions we might be able to ask. So one question could be around, do penguins with longer flippers are those wings that you see? Penguins can't fly, they are technically birds, and perhaps they wish they could fly, because it does look like it's pretty cumbersome for them to move around. But if you've seen penguins in the zoo, etc., they love sliding on ice and snow. It's and then these flippers can be used in water, and penguins can really pick up speed when they swim, right? So we want to know, do penguins with longer flippers weigh more or less than penguins with shorter flippers? So in essence, we are trying to understand, what does the relationship between flipper length and body mass look like, right? Now going back to this picture, right, we're talking about these flippers right here, right? These little wing-like things. And we want to understand, what is the relationship between flipper length and body mass, right? So you can see that this Gentoo penguin, this looks like it's bigger than both of these on either side, and maybe the flipper length has something to do with the body mass, right? So that's where we're headed with this. So this is a more articulate question. What does the relationship between flipper length and body mass look like? It's like saying, what's the relationship between X and Y, where X is the flipper length and Y is the body mass, right? So we can try to understand from this data, is the relationship positive? Is it negative? Is it linear? Is it nonlinear? And when we state the question in this way, that is a more articulate way of stating a research question, okay? Even though you might start with this, you might wonder about this, but then you want to state the question in X and Y terms, okay? We can also try to address some other attributes that might be present in the data. We might try to understand, does this relationship between flipper length and body mass vary by the species of the penguins that we're talking about, okay? And does it vary by the island where the penguin lives? So there's many ways to sort of skin this cat, and you can try to always, whenever you're looking at the data, try to think about how you might want to use those variables. Now, the first goal that we would have in this lab is to produce this scatter plot. And the reason why I'm showing you this is because it's nice to know always what we want to achieve with the data. What should the data, I mean, you may not know when you are working with the data set what the chart might look like, where the scatter plots might go, whether it's a positive relationship or a negative relationship, right? Whether it's an upward sloping line that you could fit through these points, or a downward sloping line, or a curve. You might not know that, but you still would want to always visualize what you want to put on the x-axis, what you want to put on the y-axis, how do you want to color the data on the chart, what should be the ink on the chart, etc. Right? So it's always good to look at the data and always think about, so look at the data set and always think about, what is it that how would I want to visualize this data? That's a very good way of going about it. So in this particular exercise, we're going to be producing this scatter plot here, and we are going to recreate this plot step by step, okay? So we're going to start with a blank canvas, okay? And notice in this graph here, we have flipper length in millimeters on the x-axis. We have body mass, which is on the y-axis, which is what we'll remember. And we can see that there is a scatter plot here. And what is distinguishing that scatter plot is that it is by the species of, it is colored, and even the shapes are determined by the species of the penguins, which are just 3 species in our data set, okay? And then there's like a line that's fit through these data points, through the scatter plot here. And there is some title to the graph, and there's some subtitle to the graph, okay? So that's what we want to produce, and it's good to have that at the back of your mind. Now, when we are creating a chart, we are going to be using the ggplot function. This is a very powerful function that comes from the ggplot2 package. And as long as that package is loaded in R, you can use this function, okay? Now, GG in front of plot stands for the grammar of graphics. So this is important to note. And this is the most powerful plotting function that is available to us, okay? Now, when we are working with ggplot in R, it's important to remember that there are 3 essential elements that we want to specify and supply to R, so that R can understand where are we headed and what do we want to do with the data and what type of chart we want to produce. Okay, so the first thing we want to tell R is what data are we using, right? So it's like telling ggplot, giving the function some data to use, and then we want to supply it some aesthetics. So we want to maybe tell R, here's what goes on the x-axis and here's what goes on the y-axis. Or here is how I want to color the species, or here's the shape of the species on the graph, right? So those are the types of aesthetics, and there are plenty of aesthetics that you will discover as you practice more with ggplot, but for now, this is what we want to know. Okay, so the data followed by the aesthetics, and then we want to tell R about the geometry that we want. So this is to tell R what is the visual element that we want to use in the data or in the plot. Okay, this is like, do we want a scatter plot? Do we want a bar chart? Do we want a histogram, etc.? Right? Do we want a density plot? All of those things are geometries. Okay, so let's start with understanding how do we write the first step. So how do we create this empty canvas in R? This empty canvas is a result of just using the ggplot function and then within parentheses saying data equals penguins, right? So penguins was our data set, and this is all of what we are saying. So let's copy this. Let's go back to R and let's see what happens if we do this. Okay, so let's say we put in ggplot2, okay, and we said data is equal to penguins, and we ran that. And notice there is a blank canvas that appears in under the plots tab here. And this is happening because R doesn't really know what to put on the x-axis or what to put on the y-axis. It really doesn't know. This is just an empty canvas on which you can start painting. Okay, so let's try to add some mapping now. Okay, now mapping would mean that we want to have something on the y-axis and something on the x-axis. Okay, so let's take a look at what we might be able to do. So let's say copy this, go back, and I will explain what we're doing here. Okay, so let's say we paste this line of code here, and now instead of just having data equals penguins, we're saying comma, right? So don't miss that comma. And then we are specifying our mapping. So we're saying mapping equals, AES stands for aesthetics, and then we're opening the parentheses and we're saying X equals, okay, so you're going to specify what that column name is or what that variable name is that you want to go on the x-axis, comma. Similarly, you're going to do that for the y-axis, right? So we know that y should be body mass, right? And when we look at the penguins data set, we can see that flipper underscore length underscore mm is the x-axis, right? Which is what we wanted. And then y is the body mass, right? Which is also something we have in our data set. Okay, so if we do this, if we run all of this code, we can see on the right hand side that R hasn't plotted anything, but you can see that there is the x-axis is specified and the y-axis is specified. Okay, so let's go back and try to understand how can we add the geometry. So remember that I said that there were 3 elements. We have supplied the data, we have supplied the data, we have supplied the aesthetics, and we want to now supply the geometry, right? So let's add the geometry. And what does that mean? Geometry would just mean that we're gonna just add this little line of code here to what we already did. We're gonna say plus geom underscore point. So we're gonna see how that works, okay? So let's say we said plus geom underscore point over here. Remember that the plus sign is used always at the end of the line. So remember, if we are in line number 18, this is coming at the end of that line. Okay, if I move this up, for example, again, the plus sign has to be coming towards the end of the line, not in the next line, because if I run the code like this, it's not going to work. Okay, let's try running this. Look at this on the in the console. It says there is an error in plus, right? Cannot use plus with a single argument. So what we're going to do is move plus back here and then run this, because now we want to keep it towards the end of the line always. And if we run this, this is going to run beautifully. And we can see that now we have all of our points or observations plotted on this chart. Okay, so it seems like we're moving closer to what we wanted to achieve. Okay, but we still need to dissect this by species, and we still need to label our charts, and we need to still fit that line through these points. Okay, so let's go back. Now we've understood how to add that geometry by just writing geom underscore point and empty parentheses, which is just going to create this scatter plot here. Right, so geom underscore point, if you wanted to write this down, this is going to create a scatter plot, right? That's what you use this point for, right? It's sort of intuitive that we're using geom underscore point to construct a scatter plot, because these are all points here, right? These are just x and y coordinates, if you think about it, from flipper length and body mass. So going back here, if we saw that chart, this type of chart, we can try to understand what is the relationship between flipper length and body mass. So we can say that the relationship appears positive in general, and that, what does that mean? This means that as the flipper length increases, so does body mass, right? So overall, or on average, we think that this relationship is positive. It also seems like the relationship is positive, right? It has a positive slope if you were to fit a straight line through these points. And this relationship is linear, which means that the points are clustered around some kind of imaginary line that you can think of that you fit through this. And it seems like the relationship is moderately strong, right? The points do not seem like they're scattered all over the place, but it does seem like they're clustered around that imaginary line. Okay. Now be careful. Sometimes ggplot2 is going to give you warning messages if it's dropping some data. Okay. We can go back and see. I'm not sure if it gave us anything. Yeah, it did give us a warning message here, which is important to note, that it did remove 2 rows containing missing values or values that fall outside the scale range. Okay, so we can always go back and see. Notice here itself, right? We have an NA value. So we can keep this NA value. It's really nice that ggplot2 actually tells us when it's dropping values and how many values it's dropping. So like, it'll drop 2 rows of data. So maybe there's another NA somewhere, maybe it's in flipper length, or maybe it's in body mass, right? So here's one. We don't have, on the Adelie penguin, we don't have this data, right? But there's another penguin somewhere where we don't have this data. So that's why those 2 rows were dropped. But there's different ways of dealing with it. We'll get to that later. So let's go back. Now the question for us is, maybe we're pretty convinced that this is the relationship between flipper length and body mass, but there are other variables that could influence this relationship, right? It could be that this relationship can vary by the species of the penguins that we are talking about. Okay, just before I get into this, this is just for your knowledge. It is important for you to know. Let me. It seems like this is. Okay, so basically the idea here is that you now know, this is sort of recapping, that you need to supply the data. This is the data set that you want to plot. You want to supply the aesthetics to R. These are the scales on which we want to map our data. We also want to supply the geometry, which is telling us about the visual elements used for our data. And then there are other types of elements, right? There are a total of 7 elements, which we can get to later, but these are the 3 that you need to know for now. There's a lot you can do with just data aesthetics and geometries, which we will talk a lot about in this video. And so we're going to be talking about how to color, to add color to our aesthetics, like all the points that you're seeing, which are just black right now. You can add color to that. You can change the size of that. You can change the transparency of the data that you're plotting. You can change a lot of different attributes, right, in the aesthetics, in the AES, anything that follows the AES parentheses. Similarly, when we talk about geometries, we don't just want to limit ourselves to a geom underscore point, which is going to give us a scatter plot, but we have the option of creating a line plot, like a time series plot. We can create a histogram, we can create a bar plot or a box plot. So we're going to explore these today, but there are many other elements like themes, statistics, coordinates, facets, even facets we'll talk about a little bit, that you can work with to change the way your graph looks. So once you understand the basics, it'll be really interesting to use all of these together. Okay, moving on. If we want to understand, does the relationship between the flipper length and body mass differ by species, we can really do that easily by changing the aesthetics. So if you add additional aesthetics in your plot, that can tell another story. Okay, there are different types of aesthetic mappings. So far, you've seen that you can specify what goes on the x-axis and on the y-axis, but you can, like I mentioned here, you can change a lot of these things here in aesthetics. Okay, so that's what we're gonna do. And so if we want to achieve a graph like this, for example, we are going to notice here that the color of these points, they've gone from black to red, green, and blue, and these are corresponding to the species. So the only thing we want to change in our code is that within aesthetics, right, within the AES parentheses, right after you've specified what goes on the y-axis, you're going to put a comma and you're going to say color equals species, okay? And you're going to close the parentheses. Everything else remains the same, okay? So how do we do that? We can just, all we're doing is adding this, right? So let's just go back to R and make sure we can do this. So what we're doing here is after body mass, we're saying comma, and we are saying color equals species, okay? So if we run this code again, notice on the right-hand side, we have produced a scatter plot, which is now distinguishing between the relationship, distinguishing the points by species, but also telling us that there might be a different type of line that you can fit through the blue points, the red points, and the green points. Okay, so let's go back. This is what we're doing. Now you can add different types of layers. Now remember the plot that we wanted to achieve, right? So that type of plot can be achieved if we added different geometries to those observations that are on our graph. Okay, so in addition to a scatter plot, which can be achieved by geom underscore point, we can also add a line, we can also add a smooth curve, etc., etc. Okay, so let's say if we wanted to insert the line of best fit, if we wanted to achieve something like this, where there's a line that's fit through the red points, there's a line that's fit through the green points and the blue points. What is this line telling us? Basically, this line is going to minimize the sum of squared residuals, right? The distance from this line, right, is the residual, and we are minimizing the sum of squared residuals, and that's what the line of best fit is, right? So the way to achieve this is if we were to keep the code exactly the same, but right after geom underscore point, we're going to add a plus sign, and we are going to say geom underscore smooth, and we are going to give it a method which is equal to linear model. Okay, so LM should come in quotes. And so we can try this on our own. Let's copy this and go back to R to see if we are able to achieve this. So we're going to do a plus sign. Remember, plus sign comes towards the end of the line. And then we're going to run this entire code. And notice on the right hand side, now the graph is populated with these the lines of best fits based on the species of the penguins that we are working with. Okay, so let's go back. Now we understand how to work with this. Now, this is an important point that I'm going to make between how do we create a chart that looks like this on the right hand side, and what's the difference between this chart on the right and this chart on the left. As you can see visually, we have created so far these three lines that are distinguishing the line of best fits for these different species. But what if we did not want to distinguish between those three lines? Maybe we only want to have the scatter plot colored by the species, but not that line of best fit. We want to keep the data set or consider the data set as a whole and not make that distinction. This is on the left panel. On the left panel, we have something that is basically applied to aesthetics globally. So whenever you're specifying something in ggplot, let's say you specified species in ggplot, then whenever you try to fit a line and you add plus geom_smooth, that is going to apply to all the layers of the species. Okay, so in the code, what does that mean? In the code, that means if you're coloring by species within the ggplot command, within aesthetics, right, then you will have three lines that will be produced, right? When you're trying to do geom_smooth, it's going to take a look at what is being specified in ggplot, okay? And it's going to apply that line to all of the layers that are available here, okay? So we were coloring by species, so that's what it did. However, to achieve something on the right hand side, if we look at the code, notice that the color equals species is not specified in the aesthetics here, but it is specified in the aesthetics that are within the geom_point function, okay? So this is known as applying something locally. So if you want to apply only to one particular layer, you're going to specify that in the geom later on, okay? So this is a distinction that you might want to keep in mind, that if something is applied to ggplot2, that's going to apply to all of the layers that you're specifying. But if you want that to be applied to only one layer, you're going to specify aesthetics and the color species not in ggplot, okay, not globally, but you're going to do that locally over here, okay? Okay, so moving forward, one of the things we want to keep in mind when we are creating plots is that sometimes the way we create plots might not be accessible to everyone. And mapping the shape or the color of the species of penguins may help people who are colorblind. And sometimes also using a colorblind friendly palette will help them look at the chart. Okay, so what does that mean? Usually color blindness is, when you have difficulty or you find it challenging to distinguish between the reds and the greens and maybe even the blue colors. So it's good not to use those colors and use some other colors, okay? But let's take a look at how we might want to adjust our graph. So here are some colors that are a little bit different than your red, green, and blues. Well, there is blue here, but not this blue. The way we can try to address accessibility is that we can notice in the graph, we are also altering the shape of these observations that are plotted. So we have black circles, we have orange triangles, and we have blue squares or sky blue squares. So the way we can do that is by specifying in geom_point, right? We can specify that the aesthetics we want to use is we want to color the species, and we also want to give us unique shape to each of the species. So that's what's happening. So if in case somebody is colorblind, they will be able to look at these different shapes and still be able to identify the differences. Okay. The other thing that's happening, as you can notice, is that the colors of these observations are changing in our plot. So for that, you're going to add a plus sign, and you're going to say scale_color_colorblind and empty parentheses after that. Okay, and this is going to ensure, let's copy this and let's just make sure we can see this in R. So if we try to plot this, if we run this, now we can see that that both the color as well as the shape is determined by the species. So if you are somebody who's fond of color, you can look at it and make sense of it. But if you were colorblind, then it's very helpful to have these shapes associated with it. Okay, and then also the colors of these observations are friendly for colorblind readers. Okay, so moving on. Notice we wanted to, now we're very close to what we wanted to achieve, but we still don't have a title or a subtitle. We also don't have the y-axis or the x-axis specified correctly, right? So what we can do is using the labs function, we can add some very readable and understandable labels to our plot. Okay, so notice here in this chart, this is what we want in the end, right? We want that species should be specified with a capital S. On the x-axis, we should be displaying flipper length in millimeters, not just that variable name that looked kind of ugly. And also, we want to specify the y-axis, which is body mass in grams. Okay, so it's nice to always write down what the variable is, and in brackets always have the units, as well as write the title. So maybe you want this to be body mass and flipper length. That's what we're talking about. And then the subtitle can add more information or give somebody more information about what is going on. So this is telling you that these are dimensions for the Adelie, Chinstrap, and Gentoo penguins. And this nice legend also tells us which one is which. Okay, so in order to do this, to achieve this, what are we doing? So once we have specified after geom_smooth, we're going to add another plus sign, and we're going to open the labs function here. And we're going to specify that the title equals, in quotes, body mass and flipper length. And then we're going to add a comma. We're going to add the subtitle, which is the dimensions for these penguins. Again, when you open quotes, remember you have to close the quotes, and then we're going to add a comma. We're going to add what we want our x-axis to be, again in quotes, right? These are all character vectors. y-axis, again a comma, and then we're gonna say the color scheme is basically species, and the shape is also species, right? So this is going to take care of what our legend looks like, okay? So maybe, you know, you didn't really need to alter it, but this is a capital S, which looks better over here. So now we have achieved what we wanted to achieve, and this graph looks exactly like what we wanted. Now, remember that, you know, in the code, I've been telling you to write, in this code, for example, here, I am telling you to write data equals penguins and then mapping equals aesthetics. This code is going to work perfectly fine if you got rid of data equals or mapping equals, okay? So let's just take a look here. If this is the code you were writing, remember I said you can get rid of the data equals. So I'm not writing data equals penguins, but I'm just writing penguins, comma. I'm not writing mapping equals something, but I'm writing aesthetics only, or AES, and then specifying things. This code is also going to work. This is just one way of making sure that the way you're writing code is concise. Sometimes there's just way too much text and you don't want to write extra, which is fine. But if it helps you, you know, when you're starting out in R and you want to specify that this is your data equals penguins or mapping equals aesthetics, please go ahead and write that, okay? But this is just to tell you that there is another way of writing this code in a concise way, and this is going to work just fine, okay? Finally, you could also achieve everything if you were to specify your data set before the pipe function, which we'll get into later. But notice that now the ggplot function does not have penguins written here, and it just starts with aesthetics. This is because you have already specified what the data set is. And this pipe function basically takes whatever is on the left-hand side and then says, well, like, you know, if you had to read it in English, basically it's saying, take the penguins data and then do this, right? And then do the plotting, okay? So this is how you might want to read the pipe function. It is like reading in English. Take this on the left-hand side, then do this on the right-hand side. And then if you keep adding pipes, then you're just giving it more and more commands, okay? So this is how it's gonna work. So in the next video, I'm going to be talking about visualizing distributions.